Hundreds and hundreds of plants are grown every year, traditionally from seeds to supply the main industries around us. This production system has been with us since the very beginnings of agriculture itself. But with rapid population growth and technological advancement, a question arises. Is there a way to obtain hundreds or even thousands of genetically stable plants from a single one? And if so, how are these mechanisms used today? Let's see. Large-scale food production increased, more and more quality standards began to emerge. Markets started offering products with subtle improvements that consumers could notice and prefer, often at a higher price. Take a look at these two types of strawberries. One may have been grown conventionally in an open field, while the other was produced inside a greenhouse. Protected from insects and harsh weather, cultivated above ground to avoid interference from other plants, and supplied with a carefully balanced nutrient program throughout this growth. Which one do you think was more expensive to produce, and which one will you buy? The answer seems obvious, right? One appears longer with a more uniform and intense red color compared to the other. However, the real difference is not where they were produced, but what they are. In this picture on the left, we have a variety called Red Samantha, and on the right, Red Syra. That variety on the right is earlier. It produces fruit in less time, making it suitable for regions with changing seasons. The variety on the left prioritizes traits highly appreciated by consumers, although it takes longer to produce. The decision of which one to grow depends on location, available resources, market demand, and many other factors. However, regardless of the choice, as producers, we expect all the plants we sell to be identical, available in large quantities, and free from diseases or pests. Although this may seem difficult, several techniques allow us to produce more plants in less time and space. Ideally, what we want are cloned plants that observe exactly the same characteristic as the mother plant. Naturally, some crops produce structures that allow clonal propagation, such as cones, bulbs, towers, or runners, as in strawberries. However, these processes take time, and it is difficult to obtain completely uniform population. An alternative technique that facilitates the process is micropropagation, and now we'll see why the name fits so well. Imagine you have a 7 cm shoot from a plant. After proper disinfection and working with several materials, you cut it every 2 cm, obtaining 3 segments, each containing at the last one birth. If each of these segments is placed in a jar containing the key nutrients required for growth, within a few weeks each one can reach the size of the renal shoot. If we repeat this process twice, we will obtain 9 plants, and 3 times 27 plants, and 10 times nearly 60,000 plants, starting from a single shoot. And the best part, all resulting plants are genetically identical. Since the process is carried out in a laboratory under controlled conditions, with proper care, they will be free of diseases and pests. Of course, in practice it's not quite that simple. But this essentially what happens in plant tissue culture laboratories, commonly known as in vitro culture, because the process takes place inside glass containers, and the plants must later be climatized before being transferred to the field. Although most plants have the potential to multiply this way, each species and even each variety requires specific adjustment. The responses vary depending on disinfection protocols, nutrient composition, light, and humidity conditions. Because this technique requires specialized equipment and controlled environment, it is typically reserved for species that do not easily clone themselves, and that are economically valuable, such as blueberries today.
Another important application of micropropagation is virus elimination. In this case, a specific section called the meristem is excised, a region that may not yet have been invaded by the virus. Conservation efforts also use these techniques to preserve biodiversity, giving endangered plant species a second chance to survive. Even as new application and adaptation continue to emerge, the first look at micropropagation reveals the immense potential of the technique, showing us how even the large field we can imagine may begin with a single plant.